Welcome to Interface. Today we have with us Dr. B.K. Tyagi, eminent scientist, medical orthopodologist, director retired recently from Center for Research in Medical Entomology, Indian Council of Medical Research. We'll be talking about public health challenges on dengue and chikungunya with special reference to Kerala. Welcome to the show, sir.
Thank you very much, Dr. Pushpalatha.
So, I would like to know about the common species of mosquitoes, spread epidemics and their life cycle too.
Well, there are over 3,500 species of the mosquitoes spread all over the world, and India contributes almost 400 species. There are many species which are involved in the transmission of vector-borne diseases, and in India the major infections include malaria, filariasis, dengue, chikungunya, and certain other infections which are transmitted by non-mosquito insect species and their allies. As far as dengue and chikungunya are concerned, which have been prevalent in the state of Kerala for quite some time now, the two mosquitoes which are very important are Aedes aegypti and Aedes albopictus.
Sir, could you explain the cause of infection, especially dengue?
The infection of dengue is caused by dengue viruses, which are of 4 different types. These viruses are transmitted to humans by the bite of the mosquito, which is also infective. When the mosquito bites with the infection to a person, the virus is inoculated into the human system, and the virus then goes for a long time into the human system, and then it finally comes to the peripheral blood vascular system, and that is the time when a person starts having high fever. During this high fever time, the mosquito, if it bites a person, the person will be the source of infection to mosquito, or in other words, the mosquito will pick up the virus from the human.
Sir, how is it different from the infection of filariasis?
Yeah, filariasis is a kind of infection which is caused by a group of the worms. The worm which is very prevalent in India causing lymphatic filariasis is Wuchereria bancrofti, and the infection of Wuchereria bancrofti is brought about by a mosquito called Culex quinquefasciatus. This is entirely a different mosquito than those of the dengue mosquitoes, and the other type of the lymphatic filariasis, which is caused by Brugia malae, and the type of infection which is common in Kerala and certain other pockets of the country, is transmitted by, along with Culex quinquefasciatus, also by a group of mosquitoes called Mansonia mosquitoes, which are rampant in Kerala.
Yeah. Sir, malaria also was very prevalent olden days, but now it's not that much problematic, but I would like to know about malaria also.
True, malaria has been always the #1 vector-borne infection, had been causing all over the country a lot of mortality and morbidity. Fortunately, due to good line of anti-malarials now available and good anti-malarial campaign done during last couple of decades, malaria in the country is now reduced to less than 2 million cases. Malaria is a kind of infection which is transmitted by another group of the mosquitoes. These mosquitoes belong to the genus Anopheles. There are about 10 such species which may transmit malaria in the country, of which nearly 5 and 6 species are said to be the primary vectors, because wherever they are the #1 to transmit infection. Of these vectors of malaria, 2 mosquitoes are noteworthy. One is Anopheles culissifacis, which in the country is responsible for nearly 65 to 70% of the malaria transmission in the country. The other mosquito, which is a mosquito of the urban environment and is responsible for transmission for roughly 12 to 15% of the malaria in the country, is Anopheles eastephaneae.
Okay. Sir, okay, we have to control disease means we have to go for control measures to control mosquito vectors. So, what are the common control measures as to control the vectors, disease vectors?
Absolutely true. In most of the cases of vector-borne infections, the control is basically involving insecticidal treatment to either the places where the mosquitoes rest or where the mosquitoes tend to breed into the water bodies. In case of malaria, indoor residual spraying by certain insecticides had been tried out during the last 5 to 6 decades, has been very successful, particularly in the time of 1940 late and 1950s and 60s. DDT was very successful.
Yes.
However, once the mosquitoes developed resistance, other groups of the insecticides came into picture, and currently the group comprising pyrethroids are effective, and they are being used in certain situations where the mosquitoes have developed resistance against older insecticides. The pyrethroids are proving very effective. However, because the indoor residual spraying also has certain technical constraints, a new technology involving impregnation of the sleeping bed nets, and the whole thing is called as impregnated bed nets, long-lasting impregnated bed nets, is also now being made available to be used by the people, and this is said to be very useful because they stay for almost 5 to 6 years, and they prevent the contact by the mosquito with the person who is sleeping inside, and in this way it is reported that malaria cases had been reduced to a great extent by the use of these long-lasting
Yes, sir.
nets.
What is the status of biological control in using mosquito control?
Biological control is a very important method to control. There are instances where biological control methods had been found very useful, particularly in case of malaria. We have the example of malaria control in Gujarat by integrated methodologies involving the fishes in large open body of waters where malaria mosquito were breeding, and these fishes are very specific fishes trying to feed on the mosquito larvae as a preference, and this greatly reduced the mosquito breeding into open water bodies and brought a great change into the malaria scenario in many parts of Gujarat.
Yes.
Similarly, I would recall a very important experiment which was done in Kerala in 1980s when Brugia malai lymphatic filariasis was rampant in Kerala. It was a great experiment done over here by releasing certain fishes into the ponds which were commonly available in the houses and premises of the Gambusia, etc., silver fishes, tilapia, etc., and then this has completely eradicated the mosquito which were breeding in those ponds and had brought down the cases of Brugia malai cases in the state to a very great extent.
So, nowadays many repellents are available in the market. How effective is using
repellents? Is another very good way. It's unlike the insecticides which bring about the kill to the mosquito stages. In case of the repellents, this functions on the basis of bringing about a cessation of the contact of the mosquito with the man, as a result of which the transmission cannot take place. There are many repellents available in the Indian market, but for the first time, the government department like Defense Research Organization has brought out by making a commercial opening a repellent called DEPA, which was originally invented by one of the ICMR institutes.
Yeah.
And this has now been developed into many formulations. There are many formulations of DEPA available commercially also. They are open in many outlets in the country.
Safer to use.
And safe at use. There had been all the kinds of necessary biochemical investigations had been done on the use of this repellent, and is effective from 6 to 8 hours. A very good first such a repellent which protects a person for over 6 to 8 hours and is easily available and is recommended by the Government of India for use of the military armed forces people in difficult front areas.
Sir, you have been working in Center for Research in Medical Entomology for quite a long time.
Yes.
Recipient of so many awards. I would like to know your contribution to this particular field of research.
Yeah, thank you. It had been a wonderful experience having served at Center for Research in Medical Entomology for about 10 years. I worked in many areas while I was working in CRME. My major contribution at the CRME would always be remembered for my contribution for discovering Aedes albopictus, the mosquito in Kerala, which was proved to be the primary vector of dengue in Kerala, and this has resulted in modifying some policies at the various levels within the country and other places also, in the sense that people started understanding that under certain situations Aedes albopictus can be as dangerous as the other mosquito known for its involvement in transmission, Aedes aegypti.
Yes.
And in a place like Kerala, Aedes albopictus proved its dominance, proved its ferocity.
Very great discovery, sir.
The second, another important contribution that I can always be very proud of is the discovery of the diagnostic tools for Japanese encephalitis, another disease which has been responsible for killing the children up to about 12 years of age all over the world, and in India, South India was one place that Japanese encephalitis always affected. So we had developed a diagnostic tool at CRME, and which proved to be a very good tool for making surveillance of Japanese encephalitis virus in the state, which could help in understanding the scenario of the Japanese encephalitis virus, so that we can take an action early and we can possibly reduce the vector population in an area where there could be a danger of virus transmission. So this particular technology we had transferred to the Tamil Nadu government.
Yeah, that was the two technologies you have transferred.
Yes, this was one of them. The other one also which we developed was to assist the taxonomists. We developed a kind of a kit by which the technicians and other junior scientists could use this small tool. By fixing it with a microscope, they can easily study the taxonomic feature of the mosquito vectors and flies, and they can identify in the field itself the involved vector species.
Okay, sir. What should be the role of common man to control disease vectors?
It's always a $1 billion question. We understand that the designated institutions always do their best. Nevertheless, it is very important that communities also come forward to take up certain challenges.
Yes, sir.
India is a very vast country, and unless people, particularly those who are knowledgeable people, those who are intelligent people, they must come forward and take certain responsibilities both on individual as well as community level basis. Their contribution, along with the health authorities at the state and central level, will be a great asset, will be a great utility in reducing the breeding sources of the vector populations. Take the example of dengue and chikungunya. The vectors are the same.
Yes, sir.
And the vectors breed right within and around the houses. And if the people who are living in that community are able to empty or topsy-turvy those vessels where the mosquito is breeding just every third day, there would be hardly ever any chance of the mosquito breeding into their vicinity. It's the understanding that we have to shoulder the responsibility of not allowing any of the vector mosquito breeding in our house or premises of our houses. And if that is done with a simple understanding of mosquito biology, I think we can do wonders in preventing the onset of any disease outbreak in our communities.
The very easy method to control the vector is, I think, to interfere with the larval stages.
Yes, there are certain mosquitoes where the adult control is difficult.
Yeah.
Because of their biology, because of their behavior, and because also of their development to resistance against a particular insecticide. Take the example of Culex quinquefasciatus. It's a very difficult mosquito because it breeds in all kinds of dirty, polluted waters. Sometimes boxed drains where it is difficult to spray and all that. So in those cases, control of the larvae becomes important, integral to the overall control.
Yes, sir.
So there are certain larvicides by which we can control the larval population.
And we have advantages also,
and
sir.
We have
enough duration to treat the larval stages than to the adult or the egg.
Same also for dengue and chikungunya. The mosquito in the adult cannot be so comfortably controlled because it rests outside. Dengue vector like Aedes albopictus rests completely outside the premises.
Yes, sir.
And dengue vector Aedes aegypti rests mostly not on the walls but on the hangings.
Yes, sir.
Maybe clothing, maybe other artifacts and all. So it is very difficult to control the adult. So therefore the larval control becomes a feasibility either by using a biological control or by using a chemical control. There is a particular chemical, a tamifos, which is available and which is to be used at a dose which is absolutely safe for human consumption also.
Okay.
So if this is done periodically, once in a week or so, as per the prescription of the hospital or of the health authorities, I think there would be hardly any chance of any mosquito breeding into our water utensils.
Okay, sir. Sir, even though the government have taken many measures to control mosquitoes, there will be seasonal outbreak of diseases. We always experience seasonal outbreak. What are the main factors, causative factors, to contribute this particular situation?
Yeah, I agree with you that most of the vector-borne infections are seasonal infections, in the sense the more the water bodies are available, the more the mosquitoes breed, and that also results into sudden resurgence of the infection. What is most important here is to see that in case of a disease like malaria, the program of controlling the malaria, which is to control the adult, has to be done very effectively before the monsoon season sets in.
Yes, sir.
We may not have any control on the water coming as rains, but we may always have a control on the mosquito population naturally available for breeding into that period. So if the control of the mosquito population, where the malaria is rampant, is done very effectively before the monsoon sets in, you will have very little mosquitoes to breed into the monsoon period, virtually causing no harm, no damage or no escalation into the malaria cases. Similarly, in case of dengue mosquitoes, which also comes as a seasonal infection. Suddenly in July and August, when the rains come, you find the cases are shooting up.
Yeah.
Here also, what is most important is to understand the mosquito breeds absolutely in the fresh water, the potted water.
Yeah.
So what is important here is that any water that we use to store in or around our houses, we must see to it that they are either treated, if they are not convertible, or they are not removable or emptyable, or they should be made emptied.
Yes.
If this is done, even during the period of the monsoon will remain ineffective for the breeding of the mosquito, because mosquito breeds in the potted water only.
Yes.
So if the monsoon is coming and is stretching water outside on the road, the mosquito is not going to touch that. In the pots having water, either we treat it with a insecticide or larvicide like Tamifos, or we empty them with the Tamifos once a week, or we empty them every third day. I think that will take care of it. And we don't allow any solid waste to be accumulated in our premises or nearby where we as a community live. Solid waste is a major contributor nowadays, because you can see that wherever these are dumped, these store water and the mosquito tend to freely breed into that. So we look into these aspects as a community, and there are possibilities where these can be disposed of or recycled, and we can keep our area more or less neat and clean, and the mosquito would not get. There are instances in many other countries where people have brought about such a change into their ecosystem, and the control of dengue and malaria in many countries has been a reality.
So we are continuing with integrated vector control program, sir. How we can comparable with international programs?
Yeah, this is another $1 billion question. The point here is that each of these diseases are to be dealt with in a separate manner, because their biologies are different, their control programs are different. Take the example of Japanese encephalitis, another very serious infection transmitted by mosquito, particularly to the children below the age of 12 years. Many countries today have become free from the impact of Japanese encephalitis because they have vaccinated their populations. They are vaccinated not only the human population, but also the amplifying swine population, so that the virus is completely prevented to get transmitted from one host to another host. Now, this particular mechanism seems to be very simple, and in India also the vaccination has been going on in one state or another state for quite some time. A state like Tamil Nadu, which has done it about 7 or 8 years from now, has virtually controlled Japanese encephalitis, simply because the rate of distribution of vaccine and the receiving community were very encouraging to the process. The coverage was almost 90% or 95%, so much so that that entire population was covered. Now, if this scenario is continued in other places as well, there would be hardly any possibility for Japanese encephalitis virus to get a transmission across. You take the example of dengue. There was a time when in Singapore they had completely controlled dengue, both the vector and the disease. The point here is this: there are instances both in India and across the Indian borders into many other countries where some of the vector-borne diseases had been completely annihilated or controlled. And we need to learn these lessons from how they did it, and have to possibly understand what we can transform into our own socio-economic development, and we can follow our own available resources to get rid of these diseases. It is a possibility. There was a disease called guinea worm infection,
yes, sir,
dracunculiasis, which was heavily prevalent in country in 1970s, 80s, and so forth. And then the simple method used was to filter the water so that the organisms which were carrying the parasite, the cyclops which were carrying the parasite, were filtered they were stopped to go into the drinking vessel, and then the people were drinking only the well filtered water without having the worms coming to their system, and this prevented the longevity or continuity of the parasite. Now there are examples, therefore, both in Indian states other than Kerala and in many other countries where some of these vector-borne infections had been, like in Sri Lanka recently, they have declared Sri Lanka as a country which has eliminated lymphatic filariasis. Now these examples become good examples for countries like us, where we are trying very hard to see that our country also eliminate these infections at a very early date. India has already done a great piece of effort, and in couple of years, hopefully we will be also one of those very few countries which will be declaring elimination of lymphatic filariasis from India.
Thank you very much, sir. The talk was so informative, and as a common man, we could understand what are the special, with different kinds of mosquitoes who spread diseases, and these disease vectors can be controlled by our day-to-day activities. Like, thank you very much, sir. Thank you very much for this informative talk.
So nice of you, Professor.
Thank you, sir.